We're doing experiment number 32 for Chem 104. We're looking at galvanic or voltaic cells. I use those interchangeably. And we're going to be testing various solutions as an anode or a cathode. And we have our voltmeter here, and it's attached, and here we have our red and our black, and our red is our cathode, and our black will be our anode. And we're going to look at spontaneous reactions. So remember, spontaneous reactions will give us a positive potential. Right now there's no potential because nothing is set up. These aren't even touching one another. And so we have the first two combinations that we're going to check. We're going to be using 0.1 molar solutions of copper sulfate and zinc sulfate. And remember, we're making chemical batteries. And so one is going to be the cathode and one is going to be anode, and we're going to be moving electrons from one half reaction to the other. So I have a zinc electrode that I'm going to stick into our zinc solution, which I didn't want to go too far in because I still need to get to it. Okay. And our copper will go into our copper solution. And in order to, and then I'm going to go ahead and put, I don't know which one's the anode or which one's the cathode, so I'm just going to go ahead and put our voltmeter on here, onto our electrodes, into our solution. And we still don't see any voltage. Well, if you think about it, electrons would be moving from one of these in the direction of the anode towards the cathode to the voltmeter to our zinc, but there's nothing connecting these two cells. And so what we need to do is we need to use a piece of filter paper. And our filter paper, we're going to soak it in a solution of potassium nitrate. And then I'm going to stick this filter paper with potassium nitrate in between our two cells into the two solutions. And now we see we have a voltage, but the voltage is negative. So that means I'm going to switch these two electrodes. And I'm going to put my cathode on the copper, which now, and I'm going to put my anode on the zinc, if I can get to the zinc's electrode. And we can measure our voltage, 0.884 volts, or 5 volts, 4. Okay, so that was the copper and the zinc. Now I have to make a new salt bridge for each solution. So I'm going to take this salt bridge and I'm going to stick that into a waste container. And I'm going to take my two electrodes off. And we're going to test this now with copper and magnesium. Copper and magnesium. So I have to have a manganese metal electrode in our magnesium solution. And I'm going to stick our cathode on the copper, and our anode is going to go on the magnesium. And again, we need a salt bridge. So we don't have a voltage, so I got to put this into our solution, soak up some of the potassium nitrate. Let's make sure it's in the solution. And we get a voltage. 1.693 volts. All right. We're now, again, we need to take off the salt bridge. We're also going to, another electrode that we're going to be looking at is iron. And iron, we're going to have our iron solution in our iron electrode. And I'm going to go ahead and stick, I don't know which one should be the anode or which one's the cathode, so I go ahead and stick these onto each one. Need to get our salt bridge. I might have to get some more solution here because it's pretty low. All right. Now the salt bridge is basically just some coffee filters that I soak in the solution. All right. Oh, negative, so that means we've got to swap these out. Okay. Let's make sure I'm not touching the salt bridge. And we're reading all right. Okay, 0.638. I moved it around again, let it settle. Again, I'm touching the salt bridge, so I need to make sure it's not touching the salt bridge. All right. All right, 0.638. We'll go with that. Let's take off these. All right, so that was copper and iron. And I'm going to, I'll put another solution over there. Next combination is going to be let's look at zinc and magnesium. So we look at zinc and magnesium. And I need to this is the hardest part right here. Just getting things together without, there we go. All right, salt bridge. Okay. Put our salt bridge into our solution. That one didn't stay together too well. And no electrode in there, so that doesn't help. There we go. Negative, so I gotta swap this out. There we go. So we see this is point. I keep moving things around. Let's settle. All right. Let's look at next combination is going to be zinc with iron. Oh, here's my zinc electrode. Here's my iron. Get a nice salt bridge going here. Let's wrap that up. I might actually run out. Okay. Oh, negative. So that means I need to put the all right. Okay, 0.22. All right, why did it? Oh, I was touching a salt bridge. All right, 0.217 volts. All right. Don't want to get these mixed up, but I do want to take the salt bridge out. All right, the last combination we want to do is iron and magnesium. I wish we had the same size magnesium, but we don't. Salt bridge. All right, 1.23 volts. All right. And then the last thing we want to do is another combination. And this one is going to be of our zinc with an unknown. So here's our unknown solution. And we have our zinc electrode right here. So I'm going to stick this with the zinc electrode. Now, again, we're going to be looking at potential differences between these two. And that's what you're going to be calculating. Actually, that's the wrong electrode. This is our unknown electrode. The zinc electrode was actually inside the zinc. I just couldn't see it. Okay. Now the last thing we need is our salt bridge. You have to have a complete circuit. That's why we need the salt bridge. So as electrons are moving, ions are moving in the opposite direction to keep the solutions electrically similar to one another, just like they do in regular batteries. And so here is our unknown with zinc, and that's our electron energy potential here is 0.285 volts. All right. So that is part A, where we're just looking at different chemical half reactions and seeing what the potential differences are between the two half reactions. Your goal is to write the various half reactions, figure out what metal should be at the anode and which one's at the cathode. Remember, spontaneous reactions, the E potential is positive. And then we'll be setting up for part B. Part B, we're going to be looking at our concentration cells and the effective concentration. So if you have two of the same half reactions, you would get no potential difference. But here we have two of the same half reactions. We got copper and copper with the copper electrodes that I'm going to stick into our solutions. But the only difference is that the solution here is 0.001 molar. And this solution is 1 molar. So the concentrations are different in these two solutions. So let me set up the electrodes here. So I'm going to stick these two electrodes on. Okay. All right. I'm going to get a salt bridge going here. And we're going to look at the potential difference when you have two different concentrations. Now, if they were the same concentration, you would see zero potential. But because we have two different concentrations here, we can read the potential difference. And the potential difference isn't, the energy potential is not very big. It's only 0.092, or sorry, I'm reading it backwards, 0.029 volts. Okay? And so, what we're going to do now is we can see that if you have different concentrations, you have a potential difference. We're going to take that effect. Remember, at the anode, you're doing oxidation. So you're taking the copper metal and making it into the ion, which is happening here on the left. On the right, this is the cathode, and you're taking the copper ion out of solution and plating it onto the electrode. So this electrode should be getting heavier. This electrode should be getting lighter as time goes on. So what we're going to do is now is one of the labs that we've done in the past is that we've taken copper, and if you add ammonia to it, you make this complex. So we're going to make this copper complex. So if you complex the copper, what happens is, one of your questions, so you're going to figure that out. So I've concentrated ammonia here. We're going to add 2 to 5 milliliters. And look at what happens to the potential when I add this. And look at the color of the solution. And remember that copper complex is that wonderful royal blue color. Okay, and look what happened to the potential. The potential went up by quite an amount. Okay. Now to the same cell that we made the copper complex, you had a whole chapter on solubility. And one thing to remember on your solubility rules, any compounds with sulfur, usually except unless it's with a group 1 metal or the alkali metals, don't like to dissolve in water. And so when I go ahead and add this 0.2 molar sodium sulfide, notice what happens to the solution. The solution turns this, I don't know if you can see it very well, but it turns this milky kind of blackish color. I don't know if you can see that. Right? See, it's a blackish type color now because it's formed and if, I don't know if you can see the particles of precipitate in there. Let's add a little bit more. Sorry. And what happens to the potential when we add 2 to 5 milliliters of sodium sulfite? The potential is now reading 0.526. So take this in mind and answer your questions on your report sheet. This is Chem 104, still galvanic cells, and we're doing part C, which is looking at the Nernst equation. So the Nernst equation, we are going to be looking at the cell potential difference between zinc and copper, but with different concentrations of solution. So we're going to start out with a solution that is 0.1 molar copper, and we're going to make a serial dilution. A serial dilution is where I make one solution at a time. When that solution is complete, I will make the second solution. And we want to, with these, I'm going to be using a 50 milliliter volumetric flask, and I'm going to be using this micro pipette. And this micro pipette is good to for like 1 milliliter, but I can measure out 500 microliters, which is 0.5 milliliters, so it's a half a mil. All right, so here I'm going to take a half a milliliter of our copper solution, and I'm going to stick that half mil into my volumetric flask. And now I'm going to fill this flask up to this line. You've done this in previous experiments, so this is nothing new. I'm going to fill this up until about I get to the neck, and then I'm going to use a dropper. Because if I go above that line, I have to remake the solution. And I'm not that good at pouring, so. One more half drop. There we go. Now recall this, it's hard to mix the solution with this skinny neck. So I'm going to put a cap on here, and I'm going to invert my solution 10 times. All right, so now that solution is mixed, and I'm going to label this because this is my second solution. right? Because this one's going to be my first, this is my second, this one's going to be the third, and this one's going to be the fourth. Now I need to take a 1/2 milliliter out of my second solution to make my third solution. Now my pipette will, oh, it does fit in there, so I'm going to go ahead and take it right out of here. All right. I'm going to stick that into my third solution. And then I'm going to do exactly what I did before. I'm going to add some water. And that, remember, this is a serial dilution. I can't make the next solution until I made the previous one. All right, so now I'm going to go ahead and invert this one 10 times. This is a very quantitative way to make solutions. All right, now I'm going to again take a 1/2 milliliter of my third solution and put that into to make the fourth. Okay. Now these volumetrics, remember this is 50 plus or minus 0.05 milliliters. So this is a very quantitative piece of equipment. A lot more precise than a graduated cylinder. Oops. If I watch what I'm doing, I'm spilling on the outside. All right, clean up my water here, my mess. All right, I'm going to add some water to the line. All right. Now I'm going to invert my fourth solution 4 times, or 10. 1, 2. Okay. All right, so I have my solutions. I need to stick all these into beakers now, so I'm going to put my second into my second beaker. Put it into the third. Always label your beakers so you don't get your solutions mixed up. So easy to do. So if you label your beakers, you know what solution's in which beaker. And when it comes to time to cleaning up the waste, you also know what you're doing. So I'm going to need to make a couple more salt bridges. So I'm going to do that. I need at least 4 of them, and I only have 2, 3. So let's make a salt bridge. To make a salt bridge, I'm just taking some coffee filter, and I'm just rolling it into a nice little line and creasing along the way. Again, I cut the coffee filter paper into a line, and I'm just going to roll it to make a nice salt bridge. There's a salt bridge in your batteries, and it's usually a gel that goes between the anode and the cathode. And a lot of times the anode is your the battery shell, and the cathode is a electrode that's in the center, if you've never taken a battery apart. All right, so now we're going to be testing each one of these, looking at the cell potential versus with zinc. So I'm going to look at copper and zinc, and I'm going to do each one of these solutions. And so let's look at, we need to get our salt bridge ready for each one of these. And I know we did the copper and the zinc before, but we're going to do it again so that we have consistent data here. And recall that our copper was at the cathode and our zinc was at the anode when we did this the first time. So I'm going to automatically set up the zinc as our anode, and I'm going to set up the copper as our cathode. Okay. And again, we need our salt bridge. So I'm going to stick this in here and make our salt bridge. Soak up some potassium nitrate. And I'm going to stick our salt bridge into our cell. I'm trying to make the electrodes not touch the salt bridge. There we go. And read our voltage, which is similar to what we had before. .865 volts. Okay. Now I'm going to get rid of our current salt bridge. All right. And I'm going to take our electrode from this copper solution, and we're going to wipe it down, rinse it off a little bit. All right, now we're going to trade that out and put that into our second solution that we made. Same electrode so that we have consistency. Okay, let's put our salt bridge in here. And look at and read our potential now. And remember, we were making a dilution, so we're making the solution on this side even less. So now it's reading 0.834 volts. All right. So we're going to put our third solution over. And again, I'm going to rinse my electrode so I'm not contaminating one solution into another. Because remember, I'm going to be less dilute, more dilute, less concentrated. Whoops. All right, so now a salt bridge. At the end of the two solutions, pretty good. All right. Now the voltage is reading 0.811 volts. Remove our salt bridge. And our fourth solution that we made, we'll put that in place. Got to rinse our electrode. Not a very good salt bridge this time, but we'll put our salt bridge in. And let's read our potential. Well, at least it's going in the same direction as the other ones, right? It's, as we went to more dilute solutions, the potential went down. This is reading 0.80. 8.807 volts. Okay. All right. So I'm going to take the salt bridge out so we can stop this. And that's you're going to use this information, the 4 different potentials for the 4 different copper solutions versus the same concentration of zinc. The zinc we are using is 0.01 molar. And we went from 0.01 molar, and you need to figure out the dilution that we're doing here. The dilution, remember, is 1/2 ml to 50 milliliters, and use your M1 V1 equals M2 V2. And then you're going to graph your data.